everybody gets confused. And it's because I'm still talking about these spheres, these balls, but these are different balls. These are going to be balls of cells. Previously, it was a sphere with the cells on the surface. What you're going to see at some point in this talk is balls of cells. And so the question, in a way, to be completely transparent, this question came after we started finding things with our experiments. But the question one can ask is, here, for example, we get these little groups of cells that are, yes?
Is it a ball of bacteria?
Yeah, you will see it. Yes, the answer is yes. It's a perfect sphere of bacteria. But that comes later. But here you have these little groups of cells, right? And they are cooperative. So they grow better because they're next to each other, so they can get each other's monomers and so on. But just thinking about this logically, at some point, if you would have a group like this is not going to be so beneficial, right? And the reason is that there are going to be now gradients inside. And so the things that are in the middle are going to be starving probably. It's going to be a difficult situation there because you're going to have a lot of losses here. There's going to be some area probably around here where growth is optimal. The growth rate is optimal. And so, as I keep pressing this, so which is what I tried to sketch there. And so we can also think of a model here where we have this ball of cells, which what we're trying to conceptualize is this thing, these aggregates of cells. We have these balls of cells where they produce enzymes. Then the monomers can diffuse inside the aggregate or outside. And the polymers now, it's an important difference. Please keep this in mind if you are trying to follow this. The polymers are dissolved. Yeah? In the second part of my talk, the polymers are dissolved, as opposed to the previous one where the polymer was that ball where the cells were attached. Now the polymers are in solution, and the cells are trying to get them in. They just come by diffusion. They degrade them. Monomers are released. Okay, that's the model. And here, if we do this type of things, we find there's an optimal aggregate size, which is about 10 microns, which is also the thing that we found here. But anyway, the model is a bit different. Whatever. But the optimal size where the growth rate is maximized is around 10 microns, somewhere there. And the reason is what I said, in the core, they become monomer limited if these aggregates of cells are too big. The reason why, by the way, here the polymer is dissolved is because now we're going to talk about experiments where the polymer is dissolved. So everything we do here, we always match it with experiments. So and the polymer that I'm going to talk about is in this case, alginate, which is a polymer that is relevant in the ocean as well, and it's one that we can dissolve. Okay. Yeah. So these are different strains of different bacterial species, let's call them, for simplicity, of something called Vibrio that is very abundant in the ocean. And these are experiments done by someone else, this guy here, that quantify how much they hydrolyze alginate. So basically, this thing, this rate of hydrolysis. And they're not so quantitative. They're just like they put a colony on a plate, and they measure the halo that is created. So the alginate is stained, and it's in this plate. And when the colony degrades it, then you see a halo around. And they just measure the size of the halo. But anyway, qualitatively it tells you, for example, that this guy here, 13BO1, it degrades quite fast. It produces a lot of enzymes. Whereas this guy here, where it is 12BO1, is not very good at degrading. So if you take this 13BO1 and you measure just the growth curve, these are just growth curves, where you dilute the initial number of cells. That's what you see. Okay, and then that's what you should expect to see if there is no cooperation. So again, this is one little important thing that I would like to emphasize. That's what you expect when you have just exponential growth. This type of things like this, where this is time, this is biomass, and then this is maybe this dilution, so no dilution. Then if you dilute 1 to 2, let's say, 1 to 4, 1 to 8, and so on. These are equally spaced. So if you plot, let's say, for example, what you can do is plot the time it takes to get somewhere here. Let's call this n. And the time it takes you to get there, t to n, divided by the log of the initial cell number. This is the initial cell number, the initial condition, your dilution. Then you should get something like this, where this slope is the r. But the important thing is that this is a straight line. Yeah. So I could show you that, but I think you could also do it. It's just from the equation of exponential growth, you solve for t, and you see it's a straight line with slope 1 over r actually, and and this is your variable. Now, if you have cooperative growth, this deviates. Now you start to get, this can go to infinity, because at low dilutions, then this takes forever. Right, but this is a very easy way to check if you have cooperation or not. And so in the upper one, there is no cooperation, and it's because of what I told you at the beginning. They hydrolyze so much that they're never limited by the product. Whereas this guy that is not very good at degrading, this guy is limited. And they actually, when we keep diluting, they never grow. That's why I only show these 3 lines. Okay? And now here comes the fun part. When you look at this guy here, for example, you say, okay, it managed to grow. Fine. Now, let's look under the microscope. And we stain the cells with a dye that stains DNA. And now you find the balls that I was talking about. All these balls, and it's amazing. They're pretty uniform. They form by themselves. We don't prescribe this in any way. They just aggregate and form these little balls. And in these slides, I don't have the best pictures. Now we really have amazing videos and pictures that unfortunately I cannot show you yet. But if you look inside the ball with confocal microscopy, it's just packed with cells. It's just perfectly packed with cells. Now, if maybe you have a careful eye, you will see, there are these little pockets here and there is sort of a layer outside that is about 4 cells width, and there is a surrounding thing, yeah. And this is what I wish I would show you the new movies, because you can see each cell doing things. But this was the first one that we saw that caught our attention. So there is a lot of movement inside the aggregate and nothing outside. Okay? So and this is just a clone. It's the same type of organism. No, they are doing different things, but they have divided their function. They phenotypically differentiated. So just to be clear, there is this layer of cells. There's about 4 cells here. And the cells here are moving. And they're moving in this, they have this directionality. I think that's, I'm not a physicist, but I think that's just because they are jammed. And they, like we in the metro here in Sao Paulo, we're all moving in a perfectly synchronized way. I think it's the same thing. So you can measure that, and you see that they have this flow. I know the relevance of this, well, first of all, it's a really cool phenomenon that we're now trying to study in many different ways. We are putting these things in microfluidic chambers to see how these things grow. They do amazing things. They can develop tumors. These aggregates can fuse. It's pretty amazing. And it's, one of the reasons why doing experiments is so cool is because you can find this amazing phenomena. And then, but the relevance for the context of cooperation is that these gradients that I was talking about at the beginning, they vanish now because the cells are mixing inside, right? So now these aggregates can get really big. And then after a while, the aggregates explode. So we see this now in the movies. Basically, this was just the end state. But what you see is that these things break, and then all the cells are expelled. They just create so much pressure that they push out. And then you get the empty shell at the end of the thing. And so anyway, I went from talking about cooperation to telling you about this really amazing biological phenomenon that we're trying to understand. But anyway, the main point with respect to cooperation is what I said before. We see this type of behavior in the aggregates that is non-trivial, and that enables them to grow and changes the conditions where cooperation is beneficial or not. We see aggregates where there's channeling in between that also solves the problem of the diffusion limitation and so on. Anyway, that's I think all I wanted to say for the class, because it's my last slide of my last lecture. So thank you. If you have questions.
Thank you very much, Otto. So let's take the. I think we can. I mean, can ask questions so we can finish a little bit early on Friday, and hang out longer. So time for questions. Well, while everyone thinks, I do have a question. Sorry.
Yeah.
When you showed the spatial pattern with all the clusters, have you done like spatial statistics of that, of the distribution of groups, or?
Yeah. We tried this. I don't remember anything very. I mean, there was nothing memorable.
I don't know, because it looked like a regular lattice with very well-defined regularity.
Yeah. I think we sort of gave up because it was technically difficult because these are spheres, and then you have to do a lot of imaging per sphere. You have to do the stacks, and so now we have a system where we coat a surface with the polymer with chitin, and now we can see the single cell dynamics for multiple species and other things, and in really high replication. So with that, I think that's what we would like to do. But for this, it's complicated. Yeah.
Thank you.
Yeah.
Some more questions?
The fact that the strategy that it uses is to make like a barrier that stops the dilution of the enzymes and also the substrates is directly related to the fact that they are like digesting something that's in solution. It's right. It's diluted.
Sorry, what barrier?
No, like the thing that exploded, you know, the fact that it's more rigid at the outside.
You know, I have no idea. I have no idea how this happens, why it happens. Also, I mean, well, so, I mean, you say, okay, conceptually at least, the other possibility would be that all the cells are adhere to each other in an equal, I mean, that would be the null model, so to say, where everybody is just adhere to each other, but then, you know, it just happens. And we see, by the way, this with many different bacteria. It just happens that they have this adaptation where there is a periphery of cells that are adhered, and in the middle they have behavior. Now, I don't know how that develops. Like, we haven't been able yet to see the very initial stages and where the differentiation happens. And I also know what is necessarily the evolutionary reason for why this occurs. I know it happens in many organisms, actually. People have seen this. It's just that they haven't paid attention to this type of behavior. But it's something very similar to what people see in biofilm development models, where if you open a textbook and look at biofilms, the dynamics, they will tell you that they grow on a surface and then they form these mushrooms, and then the mushrooms open and the cells come out. And I think it's the same thing. Yeah.
So, you can ask while I take the microphone.
I mean, you have measured that for different strains and different. So have you an idea of, like, I guess, like the size of these balls are like pretty regular? I mean, it seemed like pretty regular within a strain, but I guess they vary between strains.
Yeah, I don't have enough data about that. We have only recently seen that it also happens in other strains, but I don't have that information. On superficially, they look very similar.
Okay.
But I don't know. Yeah.
I was wondering about the fate of these systems, like after they explode. Could you interpret this as a strategy to disperse, or could you track the growth after this explosion?
So, I mean, if once it's dispersed, you can restart the cycle and things grow again.
Is this crust alive? Like, can it feel again after that?
No.
Those are dead.
Actually, I know, and we have been trying to figure out these cells are not dead.
But there.
So, but I have not seen what happens with this empty shell afterwards if they can sort of disaggregate and start again.
Yeah.
So far, as I've seen, they just stay there. But they, as far as we can tell, they are not dead. They're just in a dormant state. And yeah.
And could you trace if these aggregates grow faster than if they were unaggregated or something like that?
Yeah, so if they were not aggregated, they couldn't grow, right, because of the reason that. But we don't have yet, I mean, we're trying to get mutants where we can knock this off. I mean, it's going to take a while. And then we'll be able to show it, but I'm pretty sure they won't grow because of the reasons of the need to cooperate.
Some more questions?
Jacopo, I think. Oh, no, did you ask already? Yeah. Sorry. Yes? Beers?
I think those will help. Oh, there is one question there.
Hello. Actually, my question is about the first part. There was this graph that you showed with this very big variation, sort of these three sort of box plots with the three colors. Now I actually cannot exactly remember what what it was, but my question was if this variation has to do with the sort of cell state, the initial cell state of the bacteria. And if you could maybe control for that and see.
Those were so what was shown there was the hydrolysis rate per unit of mass So keeping amount of enzyme constant, amount of polymer constant, but changing the enzymes, for example, because there are many different chitinases. Maybe I should have said that actually. There are chitinases from bacillus, from bacteria, from the ocean, from all over the place. There are also different enzymes that, for all these polymers, there are different enzymes. So people have done experiments where they keep the concentration constant of the reactants, the enzyme and the polymer, and they measure a rate. That's what's there.
Okay.
Now,
so it was the enzyme, not the bacteria.
Correct.
Okay.
Correct. Now, why are these numbers so variable? Well, okay, at face value, because the enzymes are different. But I think also you're right, in the conditions of the experiments may not be the same. For example, different enzymes may have different pH optima that I don't know if they were taken into account necessarily in the experiments and so on. So that could also be a factor there, but that we cannot tell.
Okay, thank you.
So more questions? No? More beers? Well, if there are no more...
It's a quick one. You explained how it would behave if there were no cooperation, but with cooperation they start to get even more far apart as you change the...
Exactly,
is that
exactly, yeah. Well...
Because on the data you couldn't see it because they would start to die, right? For too low of a...
Right. If they needed to cooperate, then you get these things that start to then take forever, maybe, or never happen. And the reason is that then in those cases the first part of the growth, so it says like this. Here the growth is let's call this is this is the density of cells. And here is, this is proportional to density squared, whereas here it's just exponential growth. Is this, So it's just, this takes a really long time because it's quadratic.
So, more questions? Not? Okay, so if there are no more questions, let's thank Otto again.
Well, thank you.
So, it's the last talk. This is small appreciation from us, and thank you very much for coming and for your lectures. Thank you.
Thank you. feel like I graduated.
Now you can fill it with coffee. So, small reminder about tomorrow. We are not starting at 9. We are starting at 10, but we are starting. That means that we all have to be here tomorrow at 10, please. O'clock, before 10. Because as you saw today, we started 15 minutes later because no one was here, then everything goes later, and we are going to have lunch tomorrow late. Well, for me it's perfect, but you know? So please try to be on time, please. So we have a BLX talk at 10 until 11:40. Then we have a coffee break, and then we have a series of 3 short seminars on research until 1:30. And at 1:30 we go and have lunch. Okay? Perfect. It's a late lunch, but it's also a late breakfast. So